So, how does it do it? Well, this took the advent of molecular biology to really to find out what was happening. And it was found to be working on these receptors, transient receptor potentials. And at this point, the biophysicists walk in. Now, biophysicists are a strange breed, neither biologists or physicists. Strange indeed. Most of my department are biophysicists, by the way. They can be interesting people. Okay? And they're interested in basically very quantitative measurements of cell functions. And in particular, in this case, a channel, an ion channel. A channel is a protein in a lipid that allows movement of molecules, particularly small molecules, electrolytes, sodium, potassium, chloride, and calcium. And what it was found was there's these receptors called TRPV1, transient receptor potential valenoid structure I talked about, one, because it was the first one to be described, which seemed to respond to capsaicin. But the interesting thing was this channel also responded to heat. Now this is very strange. You have a chemical process which is regulated by a physical stimulus, heat. And for a long time, again, scientists didn't really understand this. And this is why we perceive the chilies as hot, basically because it's using the same receptors which we use to perceive heat, and it activates it. I'm not going to get too much into the detail, but basically what happens is this channel can be in its open or closed state. When it's open, you have a signal. When it's closed, obviously you don't get a signal. In normal conditions, the channel is closed. During heat, if you have a heat stimulus, the channel will open, allowing a signal to occur. What capsaicin does is bind to the channel, and at temperatures, ambient temperatures, allows it to become open. The channel opens and we perceive that as heat. If you have a look also, this thing is also opened by things like acid. And again, all these components, heat and acid, you associate with inflammation, tissue damage. This is why it's not surprising that the body perceives capsaicin as an inflammatory stimulus and therefore responds to inflammation. Remember, inflammation is a good thing. You want inflammation. It's only when you get things like rheumatoid arthritis and gout and all the other pathologies that it's a problem. But inflammation is good. You need it to combat bacteria and various microbes. So you start off an inflammatory reaction. Now, all of a sudden we get a paradox. This is used to prevent pain. It's used as an analgesic in things like osteoarthritis. And yet I've just told you that putting this on causes pain. Now, that's not very good, obviously. So how does something like this work? Well, if you apply it over a considerable amount of time, and these receptors are found on sensory neurons, in response to the capsaicin, those neurons release inflammatory mediators which cause the inflammation. Substance P is one. What you do is, if you keep on stimulating this, the neuron basically loses all its inflammatory mediators. They're all released. And at this point, the neurons become unresponsive. And this is how things like this, Valjex and deep heat work. They deplete the sensory neurons of the inflammatory mediators, therefore you subsequently can't feel pain. If you take it long enough, the actual neuron will die. But this is how they work. Now, just back to these receptors for a second. TRPV1 was the first one, but there's a whole host of these receptors, and they're found everywhere. And they seem to mediate our response to temperature. So here we have TRPV1. We have TRPV2, which is only activated at very high temperatures, noxious temperatures, stuff that will burn you. We also have receptors which pick up cold. So it looks like these family receptors control our response to heat. They are also found in many of them in the tongue, and how we respond to basically taste. Not so much how our response to taste, but they will potentiate the signalling. So they have no way to pick up taste like sweetness, bitterness, sour, things like that, but they magnify the response. Again, enhanced perception of flavours. Again, I'm not going to go into this, but it's a very interesting question from a physiological point of view, is how you can differentiate between cold and hot. And it's all to do with the biophysics of the channel. I'm not going to go into that. This is quite a scary slide. I'll show this to students now, you'll get panicky. But it's actually a very simple idea. Animal repellent. Again, mammals, normal mammals, like rats and rabbits, not humans, don't like capsaicin. As I said, you can treat a poultry feed with capsaicin and rats will avoid it, unless they're starving, in which case I'll take anything, but they will avoid it. Now this is a problem for plants. How do you disseminate your seeds if you can't use mammalian vectors? Well, you can use birds. In fact, it's known that birds don't respond to capsaicin. They respond to heat, but they don't respond to capsaicin. This is the first paper, it's credited with being the first paper, which looked at an animal repellent and its interaction with different species and how it had an effect on the local ecosystem. It was published in Nature. And although it looks quite confusing, it's quite simple. What they took was 3 different foodstuffs. They took an huckberry, which everything eats huckberries. We have our cactus mouse, our pack rat, and a thrasher, which is a bird. We took a non-pungent chili, and we took a pungent chili. These individuals did not me, actually. And what they showed was the thrasher ate everything. The bird just ate everything, didn't care, just ate it. The mouse and the pack rat would not touch the pungent chilies. Just left them alone.